Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing JNU PhD entrance examination for the subject biotechnology conducted in the year 2021. Okay, I have not answered certain questions from physics and chemistry, I think. The rest of it I have done. Okay, what is the approximate diameter of the SARS-CoV-2 virus? It is 120 nanometers. Answer is option 4. Then, which of the following virus did not originate or does not have a reservoir in bats? SARS, Nipah, Ebola, HIV. It is naturally HIV. Answer is option 4. Given below are two statements. Brome mosaic virus can infect humans. Brome mosaic virus is a plant virus. So statement 1 is incorrect, whereas statement 2 is correct. So that is option 2. Then next one. Which type of nanoparticles is the most toxic for cells? It is semiconductor quantum dots, whereas the other options, that is polylactic glycolic acid, silk nanoparticle, liposomes, are not toxic. Semiconductor quantum dots are toxic. Then, among the following 100 nanometer diameter nanoparticles, which would be able to carry the largest volume of cargo? So it is liposomes. Answer is option 4. Which type of nanoparticles are the most suitable as an exogenous contrast agent in magnetic resonance imaging? It is iron oxide nanoparticles. The contrast agents used are generally based on either iron oxide nanoparticle or ferrites. Then, which of the following biological entities is in nanometer range in all dimensions? It is SARS-CoV-2 virus. Then, given below are two statements. Statement 1. The behavior of nanoparticle is largely governed by the classical mechanics. Statement 2. The behavior of nanoparticles is largely governed by the quantum mechanics. In the light of the above statement, choose the most appropriate answer from the options given below. So, the behavior of nanoparticle is largely governed by classical mechanics is not correct. So, statement 1 is incorrect, but statement 2 is correct. So, answer is option 4. The cellular COP2 vesicle transport biomolecules from. So, COP2 vesicles mediate cargo transport from endoplasmic reticulum to cis Golgi. So, the answer is option 1, the rough endoplasmic reticulum to the cis Golgi. Vesicle targeting and fusion are mostly regulated via. So, vesicle fusion is mediated by binding between specific pairs of V snares and T snares on the vesicle and target membranes respectively. And RAB GTP binding proteins are required to facilitate formation of V snare T snare complexes. So the answer here is vesicle targeting and fusion are mostly regulated by GTP binding RAB proteins. Then, newly synthesized secretory proteins are localized to. So they are localized to the lumen of the rough endoplasmic reticulum. Answer is 1. Then, in mammalian rhodopsin, absorption of light triggers isomerization of the. So it triggers 11 cis retinal to all trans retinal. Then, this question was even asked for, I don't know which PhD entrance, but I think it is Hyderabad. I'm not sure about it, but you can refer other PhD entrance examinations also. You will get so many questions which will help you in practicing MCQs. I have already uploaded so many PhD entrance examination question papers. You can go through them. A neural action potential results mainly from the organized opening and closing of the. of the voltage gated ion channels. Answer is option 3. Then several internalized RTKs are degraded in. RTKs are receptor tyrosine kinases. They are degraded in the lysosomes. Answer is option 3. REC-BCD is a multi-enzyme complex stabilized by the. So REC-BCD is an enzyme of the E. coli that initiates recombinational repair from potentially lethal double-stranded DNA breaks in E. coli, which arise as a result of ionizing radiation, replication errors, endonucleases, oxidative damages, etc. So REC-BCD multi-enzyme complex is stabilized by binding of REC-A protein. Answer is option 3. Bacterial primase induces the formation of. So bacterial primase, it induces RNA primers for DNA synthesis. Answer is 2. Cytosolic pH is maintained by co-transporters, that is Na plus HCO3 minus Cl minus co-transport. Answer is option 2. Then amino acids having hydroxyl group as main functionality is or are. They are tyrosine and serine. Answer is option 2. Which of the following amino acids have more than one chiral center? That is threonine and isoleucine. Answer is option 3. You can see in the photograph here I have marked 1 and 2 for the two chiral carbon atoms in both threonine and isoleucine. Then which one of the following species can be used as a reducing agent in biological system? It is NADH. Answer is option 1. Then which of the following amino acids have heterocyclic ring? So histidine and tryptophan. If you see the photograph here, you can see they have got a heterocyclic ring, histidine and tryptophan. Answer is option 4. Then which of the following is not a reactive oxygen species? So it is triplet oxygen, which is not a reactive oxygen species. Then next one. Which of the following statements could be unambiguously shown from X-ray fiber diffraction experiment? There are 10 conformational repeating units in one turn of B form DNA, while in one turn of A form DNA there are 11 such conformational repeating units. So that can be shown from X-ray fiber diffraction experiment. Then the value of omega torsion angle in peptides is generally close to 180 degrees. Now omega torsion angle is CONH between CONH bond angle. Okay, implies which of the following? The peptide bond is generally planar and trans. Yes, answer is 1. The peptide, it is between the CONH bonds, the omega angle, so it is implies that the peptide bond is generally planar and trans. Imagine all amino acids could exist either in the D or L configuration chosen at random during protein synthesis on the ribosome. Which of the following statement would hold in that case? Now if left-handed helices they form, then they are often critical to for the correct protein folding and protein stability. So the answer will be uniquely folded globular proteins would not have existed. Yes, answer is option 1. Then which of the following is epimeric pair? It is D-glucose and D-galactose, which are epimeric pair. Which of the following enzyme is not a protein? So hammer-headed ribozyme, it is a RNA molecule, so it is not a protein. The given structure corresponds to which of the following molecules? It is that of ascorbic acid. Which of the following distance properties are always allowed for dissimilarities between gene sequences? So this comes under bioinformatics phylogenetic analysis or phylogenetic tree construction. Okay. So the dissimilarity value of a sequence with itself is always 0. That's correct. The dissimilarity value of a sequence and any other sequence is either 0 or positive real number. That is also correct. The dissimilarity value between sequences S and T is exactly the same as the dissimilarity value between sequences T and S. No, it will be different. If R, S and T denote three sequences, DRS, DRT, DST denote the dissimilarity value between sequences R and S, sequences R and T and sequences S and T respectively, then DRS is less than or equal to DRT plus DST. It should be greater than or equal to, that is triangle inequality. So option D is also not correct. So what is correct is A and B only. So answer is option 2. Then consider the assertion. In artificial intelligence based biological classification problems, use the same data set both for training and testing of the classifier is not a good practice. The assertion is correct because such practice causes underestimation of the true error rate. That is correct. Answer is option 1. Which of the following conditions have to be met for a nucleus to be observable by nuclear magnetic resonance spectroscopy? Both atomic mass and atomic number are odd. Yes. Atomic mass is odd but the atomic number is even. Yes. Atomic mass is even but atomic number is odd. Yes. Both atomic mass and atomic number are even. No, that is not the condition. So A, B, C only. Answer is option 3. Then questions 38 to 46 is from fermentation technology and I am not good with the subject, so I have not answered it. Select the virulence protein that are coded from the genes located on TI plasmid of Agrobacterium tumefaciens and facilitate the transfer of tDNA from Agrobacterium to plant cell. So VIR-D2 and VIR-E2. They function in transferring and integration of the tDNA once the tDNA complex is inside the plant cell. So answer is option 3, that is VIR-E2 and VIR-D2. Then monoploid number of chromosomes in a plant is 1/4 of the total number of chromosomes in its pollen nuclei. So monoploid number is given by X, represented by X, and total chromosome number in its pollen nuclei, that is the haploid number N. What would be the ploidy level? So first option they have given as tetraploid. So I have written it as 4x. Ploidy number is represented by X. So it is 4x, whereas 2n is for the haploid number, and I have taken a random number that is multiple of 4 as well as 2. So it is 12 I have taken, and if you see, you will find that n will be equal to 2n is equal to 12, n will be 6, 4x is equal to 12, x will be 3. So x is equal to 1 by 4n. So 1 by 4 into 6, that is not equal to x, that is not equal to 3. So x is not equal to 1 by 4n. Now second option is hexaploid. Similarly, I have taken one value which is divisible by 6 and 2. So and you will find that it is also not divisible by correct. That is x is not equal to 1 by 4n. Then fourth option will be triploid. That also is not equal to 1 by 4n. But if you see the option 3, that is octaploid. 2n is equal to 8x is equal to 24. So n is equal to 12, x is equal to 3. X is equal to 1 by 4n. So 1 by 4 into 12. So that will be equal to 3. So x will be. Then next one. Which of the following is not a true sequence of events in agrobacterium mediated plant transformation? Okay. Answer is option 3, that is pre-culture, shoot development, co-cultivation, callus formation, plantlets. So it is not a true sequence. Then which of the following is responsible for signaling of stomatal closure in response to water scarcity? It is abscisic acid. Answer is option 1. Which of the following could be the main source of eutrophication of water in lakes or other water bodies? It is nitrogen and phosphorus. Answer is option 4. Which of the following is not a source for the lignocellulosic biomass? So lignocellulosic biomass is an abundant and renewable resource from plants, mainly composed of polysaccharides, that is cellulose and hemicellulose, and also the polymer lignin. So out of this cornstarch, it is a fine white powder processed from the endosperm of the corn. So it is not a source of lignocellulosic biomass. Answer is option 4. Choose the statement that best defines metagenomics. Metagenomics, it is the study of the structure and function of entire nucleotide sequences isolated and analyzed from all organisms. So answer will be option 2, that is study of genetic material collected from a mixture of organism. Answer is option 2. The next one. For the molecular characterization of transgenic plants, tDNA copy number can be effectively determined by. So it can be effectively determined by southern hybridization experiment. Seedless bananas are triploid. Which of the following fluorescent probe is an intrinsic fluorescent probe? It is tryptophan. Answer is option. Amyloids are deposited in several neurodegenerative diseases that are rich in beta sheet structure. By using circular dichorism spectroscopy, which of the following signature peak can be used to identify the amyloid structure? It is 218 nanometers, minus 218 nanometers. Answer is option 3. Tryptophan emission spectrum of native protein shows wavelength maxima at 388 nanometers, which did not change upon unfolding in 6 molar guanidinium chloride solution. What conclusion can be drawn about tryptophan from the above observation? So guanidine chloride breaks hydrogen bonds between amino acid residues. By breaking this bond, the 3D conformation of the protein is unfolded and the aqueous solubility of the protein is greatly increased. Now tryptophan is non-polar, and if it was buried in the core, then you would not have got the wavelength reading of the native protein. Since it is present on the surface, you got the reading of the native protein as well as the denatured protein. So it is present on the surface. Answer is option 2. Molecular chaperones are a class of proteins which help the cellular protein folding by preventing aggregation. Answer is option 1. In protein sequencing, amino terminal alpha amino group is labeled with. It is labeled with 1-fluoro-2,4-dinitrobenzene FDNB. That is your Sanger's reagent. Then a 200 kDa protein was purified by size exclusion chromatography. Non-reducing SDS page shows the presence of 3 bands, that is 90, 80 and 30 kDa band on the gel. Reduced SDS page shows the presence of 80, 45, 30 kDa bands on the gel. What is the subunit composition of the protein? So reduced SDS page, it causes the breakage of disulfide bond holding the 2 subunits in 90 kDa. That is why you got 45 kDa band when you treated with reduced SDS page conditions. So it is a homodimer, that is why a single band was visible. That is at 45 kDa. So totally there are 4 subunits, that is 3 from non-reducing SDS page and 1 from 45 kDa band after reduced SDS page, which was held by disulfide bonds. So the protein is a tetramer. Answer is option 4. Then the decreasing order of hydrophobicity of alanine valine leucine is. So leucine greater than valine greater than alanine. Answer is option 1. Then in electrospray ionization mass spectrometry of proteins, which type of molecular species are present in the spectra? So if it is in the case of proteins, and proteins will be multiple charged. So they have asked in the question for ESIMS of proteins. Which molecular species are present in the spectra? It will be multiple charge because proteins are multi-charged particles. So the answer will be option 3. Then in cis peptide bond, torsion angle omega is. So peptide bond in protein structure are mainly found in the trans conformation with the torsion angle, that is omega, being 180 degrees. And if it is in the cis conformation, then the angle around omega will be 0 degrees. Answer is option 4. Mutant mice lacking in brain specific form of calcium calmodulin dependent CAMK2 will have specific defects in which of the following functions? So CA2 plus calmodulin dependent kinase 2, CAMK2. It is an abundant synaptic signaling molecule that is essential for memory formation. So the answer will be ability to remember. The next one, a drug screening cell-based assay to identify therapeutic molecules increasing CAMP levels in lung cancer cell will require. So CAMP, it is the intracellular secondary messenger, and it is short-lived and is degraded by cyclic nucleotide phosphodiesterase to 5' AMP. So what they have asked is a drug screening cell-based assay to identify therapeutic molecules increasing CAMP level in lung cancer cell will require inhibition of phosphodiesterase. Why? Because the CAMP is degraded by phosphodiesterase to 5' AMP. So if you inhibit phosphodiesterase, then you can increase CAMP levels. Then next one, overexpression of EGFR. EGFR is epidermal growth factor receptor in MCF7 breast cancer cell lines has been observed in culture. What mechanism will you target to inhibit EGFR response in these cells? So you can determine the ubiquitination of EGFR in endosomes. Answer is option 4. Then which cell surface molecule primarily mediates cell-cell adhesion mechanism for lymphocyte migration? It will be selectins. Answer is option 3. Accumulation of misfolded proteins in the endoplasmic reticulum generate endoplasmic stress. Defects in which of the following protein is responsible for this stress? It is heat shock proteins. Then which of the following inherited single gene human diseases do not involve protein aggregation in the pathobiology? So answer is adenosine deaminase deficiency. In other option, protein aggregates are involved. Then next one, which of the following inherited single gene disorders are linked to X chromosome? So it is Duncan's muscular dystrophy. Answer is option 3. Which of the following research strategies could be innovative and result-oriented in unique clinical trial design for rare diseases? So it is using patients as their own control will improve statistical efficiency. Answer is option 3. You have isolated CD4 T cells and co-cultured with macrophages in the presence of immobilized anti-CD3 antibody under 4 different conditions. So interleukin 4 and anti-IFN gamma antibody, interleukin 12 and anti-interleukin 4 antibody, transforming growth factor beta and TGF beta and IL-6. for 3 rounds to induce T helper cell differentiation and identifiable by the cytokines they express predominantly. So, how do you detect which T cell is formed on the basis of the cytokines that they express predominantly? Which one of the following is the most likely combination of predominant cytokine expression in these cultures? So, interleukin 4. If interleukin 4 is present, it will give rise to Th2 subset and it will secrete interleukin 4. So 1A will be 1. Sorry, 1. Okay, first one will be, that is interleukin 4 and anti-IFN gamma antibody. It will be Th2 subset and it will secrete interleukin 4. In the second case, it will give rise to Th1 subset because interleukin 12 is present and it will secrete interferon gamma. Then TGF beta treatment will result in the secretion of interleukin 10 and TGF beta and interleukin 6 will give rise to Th17 cells which will be secreting interleukin 17. So the answer here will be option 1, that is A will be interleukin 4, B interferon gamma, C interleukin 10 and D interleukin 17. The next one. The monoclonal antibody approved for the treatment of lymphoma is, it is rituximab. Answer is option 1. An intracellular pathogen is causing a disease that is associated with the host inflammatory response. Please suggest which one of the following may serve as an effective therapeutic approach. So since the okay, 1 minute. Depletion of interferon gamma from the system, treatment with IL-4 and IL-10, treatment with macrophage activating agent, treatment with TGF beta. Now TGF beta in option 4 and interleukin 4 and interleukin 10 in option 2 are anti-inflammatory cytokines. So that option is naturally ruled out. Whereas treatment with macrophage activating agent, it is pro-inflammatory response. So that will be more correct answer. Answer will be option 3. And depletion of interferon gamma from the system, that is also an anti-inflammatory response. Then next one. Defect in the transcription factor GATA3 will affect the release of. So expression of GATA3 transcription factor promotes the development of naive T cells into Th2 cells while suppressing their differentiation into Th1 cells. So Th2 cells, I told you earlier in the earlier question that they secrete interleukin 4. So the answer here will be option 2, that is interleukin 4. During the development of thymocytes, the double negative cells are classified based on CD44 and CD25 markers. Which cell expresses pre-TCR for the first time during developmental process? So it will be CD44 minus CD25 plus. Answer is option 2. CD19 is a marker for B cells. Answer is option 2. Among HIV infected subjects, elite controllers are. So they are HIV infected individuals who maintain undetectable viral load even in the absence of therapy. Answer is option 4. The isoelectric point of a protein is defined as the pH at which the protein has no net charge. Very easy question. Answer is option 1. Which of the following statement about the completion of the human genome sequencing in the year 2000 is false? Answer is option 3, that is all of the euchromatin sequence was finished. What is an open reading frame? ORF. ORF, it is the nucleotides of a gene that make up the codon specifying amino acids. Answer is option 2. The conversion of RNA to DNA and its amplification by PCR is called reverse transcriptase PCR. Answer is option 2. Which of the following DNA sequences can increase the rate of transcription initiation and are located 100 of base pair upstream or downstream from the genes they regulate? So they have asked for DNA sequences and it is enhancers. Answer is option 2. Mouse embryonic stem cells are used in gene inactivation experiment because they are totipotent and can give rise to differentiated cells. Answer is option 4. Which technique is used to resolve the different sizes of DNA fragments following restriction enzyme digest? So it is gel electrophoresis. Agarose gel electrophoresis can be used for this purpose. Then the online protein structure database is, so it is protein data bank. Answer is option 1. Then given below are two statements. The two sequences are homologous if they share common evolutionary ancestry. Yes. Homologous proteins almost always share significantly related three-dimensional structure. Yes. In the light of the above statement, choose the correct answer. Both statements 1 and 2 are true. That itself is correct. An example of negatively charged amino acid, it is aspartic acid. Aspartic acid and glutamic acid are negatively charged. A tool for comparing a protein or DNA sequence to other sequences in various databases is called. It is called as BLAST. Answer is option 4. BLAST is, so they have asked for the full form of BLAST. It is Basic Local Alignment Search Tool. Answer is option 3. ORF stands for Open Reading Frame. Answer is option 2. Given below are two statements. Statement 1. A small number of characteristic amino acid residues that occur consistently are called motif. True. Then motifs are typically subsets of protein domains. That is also correct. So both statements 1 and 2 are correct. Answer is option 1. A trinucleotide repeat is a sequence of 3 nucleotides that are abnormally repeated a number of times in a row. As a result, the number of repeat change as the gene is passed from parent to offspring and the signs of symptoms of genetic condition become more severe even at an early age. This phenomenon of genetic transmission of a trait that results in diseases like myotonic dystrophy and Fragile X syndrome is called. It is called as anticipation. So anticipation, it is a phenomenon in which the sign and symptom of some genetic condition tend to become more severe or it can appear at an earlier age as the disorder is passed from one generation to the next. So here a trinucleotide repeat sequence of 3 nucleotides that are abnormally repeated a number of times in a row. As a result, the number of repeat changes as the gene is passed from parents to offspring. And the signs and symptoms, yes, that is the point. The sign and symptoms of the genetic condition become more severe even at an earlier age. So this is called as anticipation. Next one. A pregnant mother with a history of genetic disease went through a chromosomal screening test of amniotic fluid at 15 weeks of pregnancy. The Giemsa stained chromosomes at mitotic metaphase showed deletion of the short arm of chromosome 5. So deletion of short arm of chromosome 5 results in Cry-du-Chat syndrome. Answer is option 2. The first living beings are considered to be single-celled organisms surrounded by an abundance of organic molecules. Probably they didn't have the luxury of experiencing oxygen and prepared organic compounds by ingesting preformed carbon molecules such as carbohydrate, proteins and lipids made by other organisms. Such organisms are called. So they are called as chemoheterotrophs. That is microbes that use organic chemical substance as source of energy and organic compounds as the main source of carbon. They are the first living beings considered to be single-celled organisms. Chemoheterotrophs. Then next one. The goal of Encyclopedia of DNA Elements (ENCODE) consortium is to build a comprehensive parts list of functional elements in the human genome. The project has been phenomenally successful in uncovering the dynamics of the human genome. However, one of the key limitations of the ENCODE dataset is that data taken from inhomogeneous population of cells with genomic instability. Answer is option 3. Then next one. The first published complete genome sequence was in which one of the following organism? So it was in CyX174 bacteriophage. Then next one. If ever you wish to find a fourth law of Mendelian inheritance that connects genotype and phenotype, the most promising space for finding the law would be, of course, biomolecular pathway. If one enzyme is mutated in the pathway, it will not give the proper phenotype. So naturally it will be biomolecular pathway. Answer is option 3. Then next one. The science of engineering biological systems from a standard inventory of parts and rational design approach is known as synthetic biology. Answer is option 4. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.